Over the past 5 weeks, you have gotten to know the major components of the central conserved apoptosis pathway: the BCL2 superfamily, APAF1-like adapters, cytochrome C, and caspases. Cytochrome C was initially identified because of its role as an electron carrier in the mitochondrial electron transport chain. And only much later was it found to also play a critical role in apoptosis, at least in vertebrates. Caspase-like proteases were initially identified because of their role in inflammation. The interleukin-1 beta converting enzyme, ICE, now referred to as caspase-1, was identified as a protease that can cleave and mature the cytokine interleukin-1 beta. Only after C. elegans Z3 had been cloned, however, was it discovered that ICE-like proteases in higher organisms also have a role in apoptosis. In contrast, APAF1-like adapters and BCL2-like proteins were initially identified because of their role in apoptosis. In the case of APAF1-like adapters, it was through genetic studies in C. elegans and through biochemical studies in mammals. In the case of BCL2-like proteins, it was through genetic studies using follicular lymphoma and C. elegans as models. Do APAF1-like adapters and BCL2-like proteins have non-apoptotic functions, like cytochrome C, for example? Do they also have a day job and a night job? At least to my knowledge, there is currently no confirmed report of non-apoptotic functions of APAF1-like adapters. However, there are confirmed reports of non-apoptotic functions for members of the BCL2 superfamily. And I would like to spend the next few minutes telling you about some of those. Many BCL2 family members localize, at least in part, to mitochondria. Those are mammalian BCL2, BCLXL, BCLW, and MCL1, and C. elegans Z9, and therefore all of the major anti-apoptotic BCL2-like proteins, the guardians. But also mammalian BAC and a fraction of BACs localize to mitochondria, and so do many of the mammalian BH3-only proteins. Most BH3-only proteins in mammals have a transmembrane domain and therefore can directly associate with mitochondria, such as BIM. Since many of these BCL2 proteins that I just listed do not only associate with mitochondria in apoptotic cells, but also in healthy cells, it might not be surprising that some of them have been implicated in various aspects of mitochondrial function. However, because BCL2 family members of course have an apoptotic functions, their manipulation always affects the apoptotic fate of cells. For this reason, studies on their non-apoptotic functions have been difficult, and in general, conclusive evidence for their non-apoptotic functions are still missing. This is a general problem with bifunctional proteins. It is often difficult or impossible to separate their functions, and therefore to determine whether these functions are dependent on each other or independent of each other. More than 10 years ago, Jean-Claude Martineau, Richard Ewell, and their co-workers, who had done very important work on BACs, noticed that the expression of BACs causes a change in the morphology of mitochondria in mammalian tissue culture cells. I briefly mentioned in one of the previous sections that mitochondria are highly dynamic and often divide and fuse. And the change in the rate of fusion and of fission can affect the overall steady-state morphology of mitochondria within a cell. What Martineau, Ewell, and co-workers found is that apart from inducing caspase activation and apoptosis, the expression of BACs in mammalian tissue culture cells causes mitochondrial fragmentation. The mitochondrial network basically fragments. This fragmentation is an early event, since it occurs even in the presence of caspase inhibitors. Furthermore, the fragmentation is mediated by the dynamin-related GTPase DRP1, which had previously been shown to be required for mitochondrial fission. What was surprising is that blocking mitochondrial fragmentation in BAX-expressing cells by expressing a dominant negative DRP1 construct slowed down the apoptotic process and delayed apoptosis. This suggested that BAX-induced mitochondrial fragmentation contributes or promotes the apoptotic process. How does mitochondrial fragmentation contribute to or promote apoptosis? A number of reports have since suggested that what mitochondrial fragmentation does is to promote the release of cytochrome C. However, how it does so is unclear. And that is in part because, remember, we still don't really understand how cytochrome C is released. That mitochondrial fragmentation in apoptotic cells is conserved in C. elegans was then shown a couple of years later by us. We found that during C. elegans embryogenesis, mitochondria fragment within about 20 minutes after the expression of AGO1 becomes detectable with an AGO1 reporter. Furthermore, we could show that the expression of a dominant negative construct of DRP can block some of the 131 apoptotic cell death that occur during C. elegans development. And conversely, we could show that the overexpression of wild-type DRP1 could induce ectopic apoptosis. How DRP1 can induce apoptosis in C. elegans is something that we have not elucidated yet. What we also found, and that brings me to the non-apoptotic functions of BCL2-like proteins, is that not only AGO1 but also SED9 was required for mitochondrial fragmentation in apoptotic cells. Remember when we talked about the fact that C. elegans does not have a BAX homolog, and about the data from the Horwitz lab that SED9 might adopt a pro-apoptotic function in cells that are programmed to die? I think this is SED9's pro-apoptotic function, the ability to, when bound to AGO1, induce mitochondrial fragmentation, which then, through an unknown mechanism, promotes apoptosis. But back to non-apoptotic functions. Based on this finding, we wanted to know whether the expression of SED9 in healthy cells has an effect on mitochondrial morphology. We found that the expression of SED9 in C. elegans, either in embryonic cells or in muscle cells in larvae, induces mitochondrial fusion and the formation of highly fused large mitochondria. And the ability of SED9 to cause mitochondrial fusion is dependent on two dynamin-related GTPases which are required for mitochondrial fusion. FISA1, the C. elegans homolog of the mammalian mitofusins, and E3, the C. elegans homolog of mammalian OPA1. Furthermore, we found that SED9 directly interacts with FISA1, which, like SED9, localizes to the outer mitochondrial membrane. Is the ability of BCL2-like proteins to induce mitochondrial fusion conserved? Interestingly, Richard Yule and co-workers had already shown that BAX and BAC are also required for mitochondrial fusion. They found that in a BAX-BAC double knockout, mitochondria were somewhat fragmented. Based on this finding, they concluded that in healthy cells in mammals, BAX and BAC are required for mitochondrial fusion. They also found that BAX and BAC promote mitochondrial fusion by activating the assembly of mitofusins, the dynamin-related GTPases that are required for mitochondrial fusion and localized to the outer mitochondrial membrane. Various reports have since confirmed that additional BCL2-like proteins such as BCL-XL and MCL1 can affect mitochondrial dynamics in healthy cells. What's still unclear is what the physiological role of the ability to affect mitochondrial dynamics is. What is the physiological role of mitochondrial dynamics in the first place? Even that is only now being systematically investigated. Mitochondrial morphology and architecture affect, to a certain degree, many, if not most, aspects of mitochondrial function. For example, changes in mitochondrial morphology due to changes in mitochondrial fusion or fission often go along with changes in the activity of the mitochondrial electron transport chain and hence changes in the ability of mitochondria to generate ATP. Conversely, changes in the activity of the electron transport chain affect mitochondrial dynamics and hence mitochondrial morphology. Based on this, it is to be expected that changes in mitochondrial morphology caused by BCL2-like proteins indirectly also affect the ability of mitochondria to generate ATP. Is this the physiological non-apoptotic function of BCL2-like proteins? To change mitochondrial morphology and therefore the ability of mitochondria to generate ATP? Changes in mitochondrial morphology probably affect other aspects of mitochondrial function as well. For example, the role of mitochondria in the storage of calcium ions. They most likely indirectly also affect cellular metabolism, something that is intimately connected to mitochondrial function. I think to unravel all this is one of the major challenges in the future. I also think that it's one of the most exciting challenges of the future. Before I move on to caspases, one more thing about BCL2-like proteins and mitochondria. There's increasing evidence that BCL2-like proteins do not only localize to the outer mitochondrial membrane, but to the inner mitochondrial membrane, and even as soluble proteins to the matrix. A publication by Marie Hartwig, Elisabeth Jonas, and co-workers a couple of years ago reported that BCL-XL is also found in the inner mitochondrial membrane, where it appears to interact with the ATP synthase and where it promotes a stable membrane potential across the inner membrane, potentially by decreasing membrane leakiness. Furthermore, Joseph Opfermann and co-workers reported that two forms exist of MCL1. One form of MCL1 that localizes to the outer mitochondrial membrane where it fulfills MCL1's anti-apoptotic function. However, a second, shorter form of MCL1 can be imported into the mitochondrial matrix through a TOM and TIM dependent process. This matrix form of MCL1 affects various mitochondrial processes and functions. It affects mitochondrial dynamics and cristae structure on the one hand, and respiration, membrane potential, the oligomerization of the ATP synthase, and ATP production on the other. As I mentioned earlier, these processes may all be functionally connected. So yes, maybe the ability to affect mitochondrial ATP production may indeed be a physiological role of BCL2-like proteins.